Hello everyone, welcome to another biology lesson. And in this class, we're going to be looking at plant structure and their function. So to start off, let's look at a leaf. Now, if we imagine a leaf, let's say we cut the leaf in half, just through here, and we took the cross section of the leaf. And now I'm going to be drawing the cross section of the leaf step by step from top to bottom, and I'm going to be labeling each layer, and later on we'll talk about the functions of each layer and each structure. So to start off, we have a layer called the upper epidermis, and that's on the top. And what this consists of is this layer is called the waxy cuticle. And it's this little black line that we've drawn at the top. And this whole thing is called the upper epidermis. Now we'll get into the functions later on. We'll just start off by labeling each part. And obviously these are all cells, so they have a nucleus. Now, under these, we have what's called the palisade mesophyll layer. And these are shaped like this. They're stacked together in this rectangular pattern right underneath the upper epidermis. And what's interesting about these are, as you can see, they have lots of chloroplasts. I'm not going to draw the whole chloroplast, but you can assume that each of these have all the same amount of packed chloroplasts. Now, underneath these palisade mesophyll layer, let me just label it. Let's just call it a tissue. We have what's called a spongy mesophyll tissue. Now these are different, as in they don't look like the palisade mesophyll tissue. They have lots of internal air spaces, and this is also for a specific reason that we'll get into later. So these don't look regular like the palisade mesophyll. They have lots of space in between them. But these are also, as plant cells, have chloroplasts in them, but not as much as the palisade mesophyll tissues. Then we reach the lower epidermis, and these also look similar to the spongy mesophyll, the upper epidermis, except they have little holes in them, which are called stomatas. So this layer is called the spongy mesophyll tissue. And this bottom layer is the lower epidermis. These are called guard cells. And we'll talk about why these are called guard cells later. And this is called the stomata. Sorry. Okay. Now, if you wanted to go further, in this spongy mesophyll layer tissue, around here, there would be the vascular bundle, and that would account for the xylem and phloem. But in this lesson, we won't cover that. We can cover that in the following lessons. So let's go over the functions of each of these. Now, if you imagine the sun coming, the sun is here, and it was shining its light rays. Photosynthesis needs light, and what the structure that carries out photosynthesis is the chloroplast, as you may have already known. And as you can see, the structure with the most chloroplasts is a palisade mesophyll tissue, this tissue here. And you can see that by all these packed chloroplasts. Obviously, if there's more, it'll be more efficient at carrying out photosynthesis. So the function of this waxy cuticle layer and the upper epidermis is that the waxy cuticle, firstly, it prevents water loss, and it does that because it's waxy. It's a protective layer on top of the leaf. It prevents the water from evaporating, and obviously water is needed for photosynthesis. And this whole upper epidermis is transparent, and this allows the sunlight to go through and reach the palisade mesophyll layer. So let's see, give it both. So they play a protective role. They prevent evaporation. They're also transparent to let light go through. So as the light goes down, now we have the palisade mesophyll tissue. And the function of this tissue is to maximize photosynthesis. As you can see, there's lots of chloroplasts, so the function would be lots of chloroplasts. But also in the way how they're arranged, they're tightly packed, and this maximizes photosynthesis. So I should mention how tightly packed they are. And yes, and this maximizes photosynthesis as there's lots of chloroplasts and they're tightly packed in this regular arrangement. And this allows most of the light to be shown on these tissues. Now, as you go down, we have the spongy mesophyll tissue, which, as we said, contains the internal air spaces. And what this does is it increases the surface area to volume ratio. Surface area to volume ratio goes up in here. And why you would need that is we can get into that later. And it also touches up on gas exchange. Now, photosynthesis needs the exchange of carbon dioxide and oxygen. So with a lot of internal air spaces, it increases the surface area to volume ratio. And this basically increases the rate of diffusion of gases, which is better for carbon dioxide to be absorbed for photosynthesis. So let's say gas spaces to create higher concentration gradients. Now, as you go down, now we enter the lower epidermis, and this contains a stomata. The stomata is where gas exchange takes place. So during the day, when photosynthesis is active, it will open up, and this allows all the gas to exchange, the carbon dioxide to come in and the oxygen to go out. And during the night, it will close. This will prevent evaporation. Obviously, during the night, there's no sunlight, and they can't undergo photosynthesis. So at night, we don't need gas exchange. So it closes during the night to prevent carbon dioxide from leaving, and it can't obviously undergo photosynthesis, so it's quite useless. So it closes, and this also helps with another factor. Plants need water. Obviously, they need that for carbon dioxide, but also they need it to remain turgid. They want water to support their structure. So if if the stomata is open, water will leave and evaporate through the stomata. So during the night, to prevent water loss, it will also close. So these guard cells, these are the guard cells. These guard cells are responsible for opening and closing the stomata. Um, the way in which these work we'll get into later, but the stomata is controlled by the guard cells. They can fill up with water or have no water, and this will basically uh, control if they close the stomata or not. So that's it for today's lesson. We covered just through the structure of the plant leaf, and how their functions are adapted um, to be efficient in photosynthesis. We'll touch up and go a bit more in depth in the next video, where we can talk about the vascular bundle, uh, such as the phloem and xylem, but also how this is more tailored to gas exchange. We can go further into how it's adapted to gas exchange, and yeah, we'll catch you guys in the next lesson.